Hi, and welcome to the 33rd Coffee Lecture Series coming from the Chemistry, Biology, Pharmacy Information Center at ETH Zurich. I'm Gina Cannarozzi, and today I'm going to talk about SciFinder N for biologists. And the question is, can I search the literature with a biological sequence? And the answer is, yes, I can. Okay, so what is SciFinder? SciFinder is an interface to the curated CAS content collection. So CAS has a number of databases with different kinds of information. For example, literature. It includes CAS references in the whole of Medline. They have CAS substances, which is registry numbers and contains 204 million substances, 69 million protein and nucleic acid sequences. They also have information on regulations, suppliers, and patents, and a whole database of reactions that they are famous for. So the nice thing about the CAS content collection is that you can move easily from one type of information for another, and that's all integrated into SciFinder, which is the interface. If I go to the SciFinder start page, which can be found at the URL above, you see this search box, and you can see here the different kinds of information that they have: substances, reactions, references, and you can search with any kind of information such as keyword, substance names, CAS registry number, etc. They have 3 specialized tools, one that's for retrosynthesis, one that's for building complex search queries using their controlled vocabulary, the CAS lexicon, and the other one is the one we'll look at today, which is searching using biological sequences. So if you go to this search sequence page, you'll see that there are 3 types of searches you can do. You can do a normal BLAST sequence, you can do a CDR sequence search, which are complementarity determining regions of antibodies and T cell receptors, and you can search by motif. So we'll look at all 3 of these in sequence. It's important to remember what sequences are being indexed. So here is what's included in their sequence search. It's the CAS registry, where for a given work, a patent, or a journal article, they only index the sequences that are essential to the work. So if you have a journal article where an entire RNA-seq experiment has been uploaded with sequences, you won't have all of those sequences in the CAS registry, only the essential ones. They also include the PatSnap database, which is a patent database, including 951 million sequences as of yesterday, December 2024. And it has an interface to NCBI, so you'll get some publicly sourced sequences from NCBI. So if I go to the BLAST page, it looks like this. I can either click in a sequence or upload a sequence. I choose the sequence type. Is it nucleotide or protein? It works with both. And I can search nucleotides or proteins, and it'll choose the correct BLAST program. I can choose to include NCBI sequences or not. And you can see I hit the search sequence and I'll get the result, but I can also set the advanced parameters, which are basically the BLAST parameters. I can choose which BLAST program I want to use for the given data type that I input, and I can choose the BLAST parameters. One thing that's important to notice here is that you can adjust the parameters for short sequences. If you have a short sequence, such as a primer, you need to use these parameters. Otherwise, you won't get any information back. So it takes a few minutes to compute the results, which is why I don't do a live demo. I'm using slides. And after 3 or 4 minutes in my search history, I'll get this the results of my search. I can click on view results there, and I get back for the sequence that I put in, which is EPO human 518 results. And these are displayed as an alignment with a graphical display of the alignment and a text alignment. And you can see over here, I get information on the alignment. I get the subject sequence, so I can reuse this sequence and the CAS registry number of that sequence. I also get the references, and this is how you can use a biological sequence to search the literature. They have a lot of filtering functionality, so I can filter the results that I get back, which looks like a list of these alignments by e-value. I can say I want to have 100% of my query sequence covered. I can filter on percent identity of the alignment and also the organism. And if you look at the organism here, you can see a lot of them are synthetic compounds, so you can also filter on that. The results can be exported as a PDF, Excel, or text. And the Excel file looks like this, where you have all of the information for each alignment, the graphical alignment, the text alignment, the parameters of the BLAST, the number of patents, the patent number, and the CAS registry numbers for each of your subjects that you've found. You can also retrieve the references for the entire collection of alignment. It will retrieve up to 1,000 references. For this collection that I have with 518 results, I had more than 1,000 literature references, so I had to make it smaller. You can do that by choosing the click boxes, as you see here, or you can adjust the filtering so that you take only the most relevant alignments. And then I get a list of literature. What's unique about this is now that you have the literature, you can search it by concepts. And concepts are a controlled vocabulary developed by CAS and assigned by professional annotators. And so this is a nice feature that you can pull out only the literature that has the relevant concepts, and this is not assigned by machines, it's assigned by humans. This function up here gives you the ability to use set functions to combine the results of this literature search with previous literature searches, and so you can do a union, intersection, or subtraction. So there's a lot of functions here to zero in on just the relevant literature and the relevant data. They have some exploratory tools that you can display the literature, where here you have the blue dots are the 4 literature pieces that I've selected, and the orange are concepts, and the green are sequences or structures. And here you can see what the literature shares. What do they have in common? What do they have that's unique? You can also visualize references, authors, concepts, substances, and organizations. And so these would look like this. If you zoom in on the 4 that I've looked at, you can see how the authors are related. And here you have the concepts, so you can see what concepts are shared and what concepts are unique. Okay, so that was the basic BLAST search. The other search functions that they have are similar, and you can use all of the same analysis tools that you can with the BLAST search. So the second kind of search you can do with SciFinder for biologists is CDR, these complementarity determining regions of antibodies and T cell receptors. These are the hypervariable regions of an antigen receptor that determine the specific binding to the antigen, and they're characterized by these short amino acid sequence. You can search up to 3 at once. So here I've put 1 on 1 and 1 of 3, and when I hit search, then again, I get the same kind of display with the text alignment, the graphical alignment, and I get a Venn diagram that shows me that 19,000 are found of CD1, 1,500 more or less of CD3, and 72 have both of these CDR sequences. This allows you to answer questions such as, has my antibody already been patented? Is my drug delivery system unique? So this is a very special feature only found in SciFinder. The third type of search are biosequence searching with motifs, and you can see here, this is a standard way of writing. At this position, I want D and E. So what this allows me to do is say I want 100% sequence identity, but at this position, I will allow a D or an E. So it's basically doing these two searches. So they have some other specialized syntax, so you can really zero in on the sequences that you want and not get back a lot of unnecessary results. They also accept the standard degenerate codes for amino acids and nucleotides. So one last thing I would like to say before I go is that CAS has a new product, the BioFinder Discovery Platform. I haven't used this one myself, but I'm looking forward to using it. It's integrated with SciFinder and the literature database, and it has more information that's relevant for biologists, including pathways, biomarkers, bioactivity, et cetera. So I'm really looking forward to seeing what we will have here. Currently, ETH Zurich does not license the BioFinder. So that's it. I'd like to remind you that tomorrow, Yoshi Dolence will be talking about Springer materials. And if you want the coffee lecture card, you can email me at cgina@ethzurich.ch and get the coffee lecture card. So I'm happy to take any questions.